Hi everybody, so we are about to start Cytogenetics I. This is the overview and a review of numerical abnormalities. This is some pretty dense material that's really text rich, so I'd really encourage you to print out these notes before watching the video and try to read through them on your own and compare the notes that I've made for you with the material written in the textbook. And then hopefully hearing my narration, which by the way is mostly also included in the notes, should help you put this all together, or at least reinforce some important concepts. So here we go. So chromosome disorders are clinical conditions in which there are microscopically visible changes in the number or structure of chromosomes. And if you have abnormalities in the number, we call those numerical abnormalities. And if you have abnormalities in the structure of chromosomes, we call those structural abnormalities. So chromosome disorders constitute a significant percentage of reproductive wastage. For example, they are present in about half of spontaneous first trimester abortions. 2, in congenital malformations. And by the way, a congenital malformation is an observable abnormality, often structural, which is present at birth. Structural congenital malformations are often referred to in the common language as birth defects. And then 3, chromosome disorders are also responsible for a significant component of mental retardation and for malignancy, that is cancer. Certain cells in your body, individual cells, can develop significant abnormalities in their chromosome number and structure, which then can result in certain types of malignancies within those abnormal cell populations. So again, chromosome disorders are a very large group of disorders that have a far-reaching range of clinical sequelae. And as a whole, chromosome disorders far outnumber those single gene disorders, all right, which many of you are actually researching for your application paper topic. In fact, there's hundreds of chromosome disorders. They are present in nearly 1% of live births and about 2% of pregnancies of women older than 35 who undergo prenatal diagnosis. So, chromosomal disorders can involve abnormal numbers and/or structures of both autosomes and sex chromosomes. The most common numerical abnormality is called aneuploidy. Aneuploidy is simply the state by which there are missing and or additional, rather, chromosomes for a particular type of chromosome. That is, there are missing or additional chromosomes for a particular type of chromosome. As a result, aneuploidy results in abnormal overall numbers of chromosomes in a genome. Okay. It's important to keep in mind that the aneuploid state will always result in physical malformations and/or cognitive deficiencies. In fact, most cases of aneuploidy result in spontaneous abortion of the embryo during the first trimester. The most common structural abnormality in chromosomes, that is a reciprocal translocation. Reciprocal translocations occur when segments between non-homologous chromosomes are swapped. It is important to keep in mind that these translocations often do not result in changes in phenotype. This is because often all of the genes in the swapped segments of the chromosomes remain intact despite being in different locations of the genome. It doesn't really matter where they are in the genome. As long as all the genes within these swapped segments are okay, then you're not going to see any changes in the overall phenotype, okay? of the person, okay, that is in their characteristics, okay, and general health. So an exception to this occurs, however, when the genes located in the regions where the segments detach, okay, from these chromosomes, okay, are interrupted or broken. Okay, so in those areas, those kind of breakpoints where these segments are actually come away and detach from the chromosomes, okay, before they're swapped with another non-homologous chromosome. If a gene is located at those breakpoints, then that gene is going to be damaged. So in general, overall, the impact of chromosomal abnormalities are highly variable and hinge around the nature of the abnormality and the nature of the specific genes that are impacted. So as a result, predicting the phenotype of a chromosomal abnormality as can be detected in fetuses via prenatal screening can be very, very challenging, or at least at this point. So let's talk about the heteroploid state. The heteroploid state is the state where the chromosome number is not normal. Okay, this is a big tent category. So if the chromosome number is not normal, you're dealing with a heteroploid state. So in humans, a heteroploid state exists when the total number of chromosomes is not 46, right? Because in a normal human diploid cell, okay, that is in a cell that has two sets of chromosomes for each type of chromosome, okay, for a total normally of 46 chromosomes. If in case it is a fact that there are not 46 chromosomes, then you're dealing with a heteroploid state. So heteroploid comes in two different categories, euploid and aneuploid. We're going to talk about euploid first. The euploid state is the heteroploid state in which the total number of chromosomes is a multiple of the haploid chromosome number, or N. And remember, the haploid chromosome number is just the number of unique types of chromosomes. And in humans, N equals 23. We have 22 autosomes, meaning the non-sex chromosomes, and then we have one set of sex chromosomes, okay, which come in two different flavors, X or Y. And then we have the aneuploid state, okay, which is a heteroploid state in which the total number of chromosomes is simply not euploid, okay? So if you're dealing with a heteroploid state that's not euploid, meaning that the total number of chromosomes is not a multiple of the haploid number, which is 23, then you're dealing with an aneuploid state, okay? So again, in other words, aneuploid describes a state in which any abnormal total chromosome number exists other than that found in the euploid state. So again, heteroploid describes the state where the number of chromosomes is not normal. Okay? In humans, the heteroploid state exists when the total number of chromosomes is not 46. Okay? If the heteroploid state exists where the total number of chromosomes is a multiple of the haploid chromosome number, or N, it is specifically referred to as euploid. More specifically, if the number of chromosomes is 3 times the haploid chromosome number, or N, a triploid or 3N state exists. If the number of chromosomes is 4 times the haploid chromosome number, a tetraploid or 4N state exists. Because in humans N equals 23, the total number of chromosomes in the triploid or 3N state is 3 times 23, which equals 69. Likewise, the total number of chromosomes in the tetraploid state or 4N state equals 4 times 23, which of course equals 92. By the way, there is a typo in the notes. I state that 3 times 23 equals 92. In fact, I meant to write 4 times 23 equals 92, referring to the tetraploid state. Another thing to keep in mind is that in the triploid state or 3N state, there are 3 homologous chromosomes for every unique type of chromosome. Okay, so for chromosome number 1, 3 homologous chromosomes. Chromosome number 4, 3 homologous chromosomes. For the sex chromosomes, 3 sex chromosomes. All right, 2 X's and a Y, or 2 Y's and X. In the tetraploid state or 4N state, there are 4 homologous chromosomes for each unique type of chromosome. So again, for every single unique type of chromosome, you have 4 homologous chromosomes. So again, if any heteroploid state exists that is simply not euploid, okay, it is referred to as aneuploid, as in not euploid. All right. The majority of the aneuploid states can either be subclassified as being either trisomy or monosomy. The trisomy state exists when there is the presence of 3 homologous chromosomes for only one particular type of chromosome. Okay? Recall that normally there are pairs of homologous chromosomes. That is 2 homologous chromosomes for each unique type of chromosome. Okay? And the most common trisomy out there would be Down syndrome, or trisomy 21, where you have 3 homologous chromosomes for chromosome number 21, but only for chromosome number 21. All of the other chromosome types just have pairs of homologous chromosomes. That is just 2 homologous chromosomes. The monosomy state exists when there is the presence of only 1 chromosome for a particular type of chromosome. Okay, for example, you only have 1 sex chromosome. You only have 1 X chromosome. All right. So let's talk about some of the mechanisms by which these euploid states come about. So first we will talk about the mechanisms by which the triploid state comes about, and then we'll talk about the tetraploid state. So later on, we're going to talk about, very shortly, that is, within this lecture, we'll talk about the clinical sequelae that comes about as a result of these different mechanisms. So the triploid state can occur via 2 different mechanisms. The most common cause of the triploid state is dispermy. Dispermy occurs when a normal egg with a haploid or N chromosome number is fertilized by 2 normal sperm, which also possess each normal haploid or N chromosome numbers. And so think about it in just terms of basic algebra. The egg possesses a chromosome number of N, which is 23 in humans. Each sperm that fertilized the egg has a chromosome number of N, again, 23 chromosomes. Therefore, the fertilized egg, or zygote, possesses a total of 3 N chromosomes, because having 1 N from the egg plus 1 N from one sperm and 1 N from the other sperm adds up to a total of 3 N. All right? If you insert 23 for N, because in humans N equals 23, the total number of chromosomes in the resulting fertilized egg, or zygote, is 3 times 23, which equals 69. The triploid state can also exist when an abnormal egg with a diploid or 2N chromosome number is fertilized by a normal sperm, which again has a haploid or 1N chromosome number. Okay? So we're going to talk about shortly about how that happens. How do we get an abnormal egg that has a diploid or a 2N chromosome number? So think about the scenario again in terms of basic algebra. The egg possesses a chromosome number of 2N, which is not normal. All right, and also n equals 23 in humans, and then therefore 2n equals 2 times 23 equals a total of 46 chromosomes. The one sperm that fertilizes the abnormal diploid egg has a chromosome number of n equals 23. The sperm has 23 chromosomes. Therefore, the fertilized egg or zygote again possesses 3n chromosomes because you have 2n from the abnormal egg plus 1n from the normal sperm equals a total of 3n. And again, if you insert 23 for n, because n equals 23 in humans, the total number of chromosomes in the resulting fertilized egg or zygote is 3 times 23 equals 69. So in contrast, the tetraploid or 4n state results from the failure of an early cleavage division in the zygote. Okay? So initially, the zygote has a normal diploid 2n chromosome complement, just like all good normal zygotes have, right? The zygote is the result of the 1n sperm merging with the 1n egg to give you a now 2n chromosome complement, okay? However, the zygote starts to enter mitosis, as was previously described in previous lectures. But the chromosomes fail to be distributed on the opposite poles of the cell, and the cleavage furrow during telophase doesn't form right. And as a result, after this kind of, abnormal mitosis is through, you still have twice as many chromosomes, but they're not distributed amongst two new daughter cells. Instead, they remain in the same cell. Okay? So you have a cell that goes from being 2n to a cell that has double the number of chromosomes, a cell that in fact is 4n. And then this embryo continues to develop from hence this point with 4n chromosomes in each cell. Now, in some organisms, okay, like the xenopus frogs, which a lot of people in the scientific world use as a model organism, or believe it or not, strawberries, the 4n or tetraploid state works very well, but in humans, not so much. Okay, it always results in a early spontaneous abortion. Okay. So the triploid state does result in the development of a little bit more viable tissue, but ultimately it also, much like the tetraploid state, will result in early spontaneous abortions as well. Okay, and we'll talk about the specific nature of those reproductive waste products right now. Okay. So the triploid state most commonly results from dispermy, where you have the fertilization of a normal egg with 2 sperm. Okay, and this results in an extra set of paternal chromosomes, right? Because the sperm, in fact, are coming from dad, right? Okay, so you have an extra set of chromosomes coming from dad if 2 sperm fertilize a single egg. And this is going to result in 2/3 of your what are referred to as partial Hydatidiform moles. And just in the next slide, we'll talk about exactly the nature of a Hydatidiform mole or a molar pregnancy. This is a specific type of Hydatidiform mole called a partial Hydatidiform mole. And we'll talk about the differences between partial and complete Hydatidiform moles shortly. But anyways, of the partial Hydatidiform moles, 2/3 of them are going to be resulting from this mechanism, as in the fertilization of a normal egg with 2 sperm, that is dispermy. Most often results in early spontaneous abortions. Okay, so the triploid state can also occur from the fertilization of a diploid or 2n egg. And you get these abnormal eggs when you have a failure of meiosis 1. All right, so that first division in meiosis 1 fails, and you're not able to cut down your total number of chromosomes from diploid to haploid, that is from 2N to 1N, like you're supposed to. So you just keep on going on through meiosis 2 with a what will become a 2N egg. All right, and the eggs are from mom, right? And so you're going to have your extra set of chromosomes from mom. They're going to be maternal chromosomes. And this is going to result in the other third of partial Hydatidiform moles, okay? Which also result in early spontaneous abortions. So now let's talk about Hydatidiform moles. A mole or a molar pregnancy occurs when abnormal development of the chorionic villi, okay, of the placenta. The chorionic villi of the placenta, by the way, are those sac-like structures which are filled with the fetus's blood, where you have exchange of oxygen and CO2 with the maternal blood flowing through the uterus, and you also have exchange of nutrients and antibodies and such occurring at that location. Well, instead of having normal chorionic villi, you have these huge cyst-like or grape-like structures called hyatid cysts. All right, and moles are classified as either complete or incomplete or partial, as the terminology was used in the previous slide. Incomplete and partial are the same thing, is what I'm trying to say. And with complete moles, you have complete absence of placental material, and then in a partial or incomplete mole, you will have a little bit of a normal placental tissue, and you might have a little bit of an atrophic fetus as well. But anyways, here we have on the right, you can see all of the hydatid cysts making up the majority of the Hydatidiform mole within the uterus. Okay. So let's talk about complete moles quickly. Complete moles, you have, again, no normal placental tissue is present. The fetus is not present, okay? Or even if you do have one, then it's grossly misformed, is what I was supposed to trying to write there in that box. And then also, again, you have the incomplete or partial moles, and they're called partial because you have the partial presence of a normal placenta, and there may be a small atrophic fetus as well. Complete moles, which weren't mentioned previously, occurs when a normal 23 sperm, okay, or 23 chromosome sperm, rather, fertilizes a secondary oocyte or egg that's missing a nucleus, okay? And as a result, the genome, okay, of that 23X sperm is replicated so that the resulting zygote still has a diploid chromosome set of 46 chromosomes with double X for the sex chromosomes. But in this case, all of the chromosomes are going to be of paternal origin, right? Because all of the chromosomes originated from the chromosomes of that one sperm. And in fact, all of the homologous chromosomes are actually identical to one another, okay? And therefore, all of the alleles, meaning all of the different versions of the genes for all of those chromosomes, are also exactly the same. And during development, that causes a lot of different problems, resulting in this complete, amorphic, dystrophic, unorganized form of reproductive wastage. Okay. Again, in incomplete moles, 2/3 occur when the secondary oocyte or egg is fertilized with two sperm, that is called dispermy, and the other third occurs when you fail to normally go into meiosis II, and as a result, you end up with retaining an extra set of chromosomes in the egg. You get an abnormal diploid egg, and you have an extra set of maternal chromosomes. All right, so that is a big picture summary, okay, of the nomenclature that we use within cytogenetics, and the major numerical abnormalities that you can run into. I guess that's all I want to talk about for now. I'm going to save the rest for Cytogenetics II. So stay tuned for that. And I guess I'll be in contact with you soon via email, and looking forward to meeting everybody in person in the next couple of weeks when we start having these writing workshops. So that's all until then, and I will see you soon. Bye.